So chapter 9 is all about cell respiration. So it's how do we take that glucose that's produced during photosynthesis and put it into a form that is usable to do work within a cell. This is a complete shocker to you, but all living organisms need energy. And ultimately, all living organisms get that energy from the sun. Primary producers get that energy from the sun and convert light energy to chemical energy, and every other organism, your heterotrophs, eat primary producers or other animals to convert that chemical energy into another form that's usable for them. As we learned in ecology already, energy flows into the ecosystem in the form of light energy from the sun and gets transferred to chemical energy in plants. And then as that energy moves up the trophic levels, most of it is lost as heat or returns to entropy in the universe. Photosynthesis generates oxygen as well as organic molecules like glucose, which we use in cellular respiration. Cells use chemical energy that are stored in glucose to regenerate ATP, which gets recycled, and it's the ATP that actually does work within the cell. I really like this diagram because it shows how photosynthesis and cellular respiration are related. Energy enters as light energy and is trapped by chloroplasts that convert the light energy to chemical energy in the form of organic molecules like glucose. So the products of photosynthesis are glucose and oxygen, which are the reactants for cellular respiration. Cellular respiration uses glucose and oxygen to create ATP, which does work within the cell, and other products of cell respiration are carbon dioxide and water, which are the reactants for photosynthesis. So the two processes are interrelated, which create a cycle for organic molecules to move through. As we talk about cellular respiration, there are two pathways, the catabolic pathways and the anabolic pathways. And in cell respiration, we are concerned with catabolic pathways, which are the breakdown of organic molecules. So this is exergonic, it releases energy. This is how we get energy to perform work in cells. There are two types. There is fermentation, which is the degradation of sugars that occurs without oxygen present. So we consider it an anaerobic or without air reaction. And we also have aerobic respiration, which consumes organic molecules such as glucose in the presence of oxygen to yield ATP. So this is aerobic respiration, meaning in air. When we discuss cellular respiration, it can include both aerobic respiration, so without oxygen, your end product being fermentation, or it can include aerobic respiration where it is within oxygen. Most of the time we're using it to refer to when we have oxygen present. So keep in mind that we can break down carbohydrates, fats, and proteins to provide energy to do work in the cell, but for our purposes today, we're going to focus on the carbohydrate glucose as we look at the reaction. So cellular respiration is C6H12O6, which is glucose, plus oxygen produces CO2, H2O, and energy, as well as some heat. So some energy is lost as heat. It's not a perfect conversion. In order to understand cellular respiration, you have to have a pretty good understanding of redox reactions or oxidation and reduction reactions. And what's basically going on is we are looking at the transfer of electrons during chemical reactions, and when you transfer them, you release energy that's stored in organic molecules like glucose. We can use that energy to synthesize adenosine triphosphate, or ATP, from ADP and an inorganic phosphate. So we're taking that third phosphate group and we're sticking it back on and forming a bond. And you'll remember from previous chapters also that ATP is used to do work within cells. So when we have transfers of electrons, we consider these chemical reactions redox, or oxidation reduction reactions. So a couple definitions that you need to know. In oxidation, your substance is losing electrons, or we consider it oxidized. This is a loss of energy because you're losing electrons. In reduction, your substance is gaining electrons, or it's reduced. And the reason we call it reduced when you're gaining electrons is because your amount of positive charge is reduced. It's becoming more negative. And this is a way to gain energy. And one way to keep this straight is through the acronym LEO, says GER. and it's GER spelled G-E-R. And Leo says GER stands for loses electrons oxidized, gains electrons reduced. So it's a way to remember that oxidation loses electrons and reduction gains electrons. This is a simple reaction to show us where the electrons are transferred and the terminology that you'd use to explain that. So in this equation, you have sodium who loses an electron to chlorine. It becomes oxidized because it's lost an electron. Chlorine becomes reduced because it gains that electron from sodium. So chlorine is reduced, gains electrons, and sodium is oxidized because it's lost an electron. And this is a more generic form of this, okay? Here X is losing its electron, so it has been oxidized, and Y is gaining an electron, therefore it has been reduced. All right, this one's a little more complicated, but now we're actually looking at a combustion reaction, which is similar to the breakdown of glucose in cellular respiration. So here we have methane, who is the reducing agent. The reducing agent is the electron donor, and because it is losing an electron, it is being oxidized. Oxygen is your oxidizing agent because it's accepting the electron and it becomes reduced. So now we've introduced those terms, reducing agent and oxidizing agent, and if you forget, here's the definition that'll help you to keep it all straight. So let's get specific about cellular respiration. In cell respiration, glucose is oxidized, so it loses electrons, and oxygen is reduced, meaning it gains electrons. So here we have glucose, C6H12O6, and a good way to remember that formula is that it is the times that you eat, 6, 12, and 6. And so that loses electrons, it's oxidized, and oxygen gains those electrons, so it becomes reduced. So in order for cell respiration to release energy that can do work, we tend to break down glucose in a series of steps rather than releasing all the energy at once. So what we do is we take those electrons that were from organic compounds like glucose, and they're transferred first to NAD+. This is a coenzyme. It is an electron acceptor. Because it's an electron acceptor, we consider it an oxidizing agent during cellular respiration. So NADH is the reduced form of NAD+. So this has accepted the electron already, and it represents the stored energy that is tapped that we can use to then make ATP to do work within the cell. Your big takeaway here is just to look at the charges and to see what exactly is going on as that electron is transferred. So this is NAD+ in its structural form, and we add to that 2 hydrogen ions, or 2 positive charges. And the reason we add 2 positive charges is because we pick up 2 electrons. We accept 2 electrons. NAD+ already has a positive charge, and if we add 2 negative charges to it, the overall charge would be negative. And our goal is to get a molecule that has no charge at all. So that's why we have to add 2 hydrogen ions, because we're picking up 2 negatively charged electrons. So your final product is NADH, which is carrying those 2 electrons it has accepted, as well as 2 extra hydrogen ions or positive charges. NADH will then pass the electrons to an electron transport chain. And so what this does is it releases little amounts of energy at each step. If you had released all the energy at once, you'd get one explosive reaction. It would be similar to taking a barrel of gasoline and lighting it on fire. You'd get a big ball flame, light heat, all that energy is released at once. That's not conducive to life. So we're going to have to release just a little bit at a time. So oxygen is going to pull the electrons down the chain, and that is going to release energy as it moves through the electron transport chain. And we use this energy that's released to regenerate ATP. That's our big goal, is to make ATP, which can do work in the cell. Because we like pictures, this is what it looks like pictorially. So if we take an uncontrolled reaction, we release all the energy that's available at once, you get this big ball of heat and fire and light, and things explode, and there's death and destruction. But if we take the energy and we release it step by step by step by step, we can release little bits of energy at a time. Because of that controlled release, then we can use those small amounts of energy to make ATP. And that's what your body does. It's not releasing all the energy at once. That would be explosive, and death and destruction would ensue. All right, so cellular respiration has three main steps: glycolysis, the citric acid cycle, and oxidative phosphorylation. In glycolysis, you're breaking glucose into two molecules of pyruvate. This happens in the cytosol, or that liquidy part of the cell. It doesn't happen in any organelle. The citric acid cycle, or the CAC, is completing the breakdown of glucose. You're breaking it apart more, releasing energy, and this happens in the mitochondria. It is not until oxidative phosphorylation do we get most of our ATP synthesis. This accounts for the big amounts of ATP that are made, and this is happening in the inner mitochondrial membrane. Keep in mind, for aerobic cellular respiration, glycolysis occurs, and then if oxygen is present, then we go through the citric acid cycle and oxidative phosphorylation. If there is no oxygen present, we go from glycolysis into fermentation. All right, so this is showing us glycolysis, where you have a glucose molecule that's converted into two pyruvates. What you're doing in this reaction is you are you're losing electrons, and NAD plus is picking up those electrons. So NAD plus acts like this carrier that takes those electrons and move them to where they're needed within the cell. So this releases just a little bit of ATP, but this is the first step of cell respiration, and it happens in the cytosol. The next step, if there's oxygen present, your pyruvates are going to move into the mitochondria and go through the citric acid cycle. Some ATP is produced, not much, and then of course our big push here is to get those electrons released from the organic molecule, pyruvate, and they get picked up by the electron carrier NAD+, and also the electron carrier FAD, which also picks up 2 electrons and 2 hydrogen ions, and it carries it to where it's needed within the cell. Our last step is oxidative phosphorylation. So this is the moneymaker for ATP. In oxidative phosphorylation, you have two parts: the electron transport chain and chemiosmosis. And it's chemiosmosis that acts like an ATP factory. It is using all of these electrons that have been lost through the breakdown of organic molecules and all those hydrogen ions that came along with it. It's using those to harness it to do work through the electron transport chain. Chemiosmosis uses ATP synthase, which actually makes your ATP from ADP an inorganic phosphate. So oxidative phosphorylation is where you get 90% of the ATP that is generated from cellular respiration. Remember, that happens in the very end of cellular respiration. You have to have oxygen in order to get 90% of that ATP, and it has to happen in the inner mitochondrial membrane. A small amount of ATP is formed in glycolysis as well as the citric acid cycle, and we call this substrate level phosphorylation. So you're getting just a tiny bit of ATP, but not the big powerhouse that powers most of your cell's functions. This is what substrate level phosphorylation looks like. You have your substrate with a phosphate group and an enzyme that literally takes that and makes a product and some ATP at the same time.